Good afternoon, everyone. Thank you for sticking around for the last presentation. I'm assured that there is coffee right after this. Following Sandy is always such a pleasure because he has such a nice presentation style and really, sets the tone for, how many of the companies that presented in this session, are really here to try and find and improve, the lives of patients. And so Abeona is a rare disease company. Of course, we are also a public company, so once again, another public safe harbor statement. But when we talk about clinical trials and gene therapy and cell therapies, Abeona is really a global leader with 3 clinical trials right now. 2 of them are leading world stage at this point. We don't have a lot of time today, but I am going to talk about our lead programs in Sanfilippo syndrome for the ABEO 102 program for MPS IIIA, as well as the epidermolysis bullosa program, which is EB 101. So here is our rare disease pipeline. Like many of the companies, we seek to go after many of the orphan product designations, both here and abroad, as well as the rare pediatric disease designations. Very pleased that we were recently granted the breakthrough designation for our EB 101 program. And now as we seek and continue to find and develop more data on some of these programs, we'll certainly be seeking more advanced designations. So to talk about our ABEO 102 program, I'm actually going to present some data up here today to show you some of our really world-leading data on the MPS IIIA program. It is currently the only program in the world that is enrolling patients in the MPS IIIA program as a gene therapy. So just to refresh, we've heard a lot about lysosomal storage diseases today. This is MPS III or 3A. There are 4 versions, A, B, C, and D. We have clinical programs for type A and type B. And again, 70% of these children do not reach the age 18. There are no therapies out there. There are, however, a number of notable failures in this space, particularly around the enzyme replacement therapies or some previous gene therapy approaches. We do have a supportive natural history study, and as many of us in the audience know, the critical nature of having a natural history study to support the clinical paradigms and the outcome measures that you're looking at. So we have 25 patients that have gone through 1 year worth of follow-up. And now we have 2 ongoing clinical trials, both here and in Europe and in Australia. So again, we have enrolled a number of patients at Nationwide Children's Hospital and look forward, the sites in Europe and in Australia are up and running. We look forward to presenting a more fulsome update at our Research and Development Day coming up next week, as well as the European Society for Gene and Cell Therapy. But part of my mission here today is to give you a little bit of a snapshot about some of the things that we're seeing. So this phase 1/2 MPS IIIA program, again, 25 patients. The important thing about these is that the natural history studies validated the outcome measures. We talk about this triangle of efficacy that we are looking at for when we see biopotency or some of the biophysical manifestations of the disease, as well as the neurocognitive changes. We have 2 dose cohorts that have enrolled and are continuing to enroll an additional 6 to 9 patients. Primary objectives of this is safety with efficacy, looking at, again, biomarkers, some organ changes, and neurocognitive changes. So let's talk about some of the data. And again, for those of you that don't know, we have 4 programs that are AV9. This is, of course, I think as was mentioned earlier, one of the, if not the best way to approach a lot of the CNS diseases. We happen to be using this as an intravenous delivery, which has been, I think, very broadly appreciated by a number of other companies, including Avexis and some others, that when you give this by an intravenous administration, it crosses the blood-brain barrier to really deliver very exquisitely and broadly across the CNS, where you're actually able to see things in the deep brain architecture where I think some of the other approaches may be challenged. So again, we're going to talk a little bit now about some of the biopotency, so looking through 1 year. So I think many of you in the audience, I think I've seen, are familiar with some of our programs. We are now seeing a dose and time-dependent durable reduction in CNS heparan sulfate. So for those of you that are not familiar with these diseases, heparan sulfate is the sugar that the kids can't break down. And this, we are showing that a single intravenous administration of an AAV vector has crossed the blood-brain barrier and is now able to act a year later, to dose-dependently reduce the heparan sulfate. So, and again, we do note that this has been a significance of p.001. Similarly, with these lysosomal storage diseases, while the CNS is often the first among equals, there is many systemic deficits. So they have enlarged livers and spleens. They have eye problems, joint problems, intestinal problems. So a lot of the times treating the CNS only is not necessarily the best approach for this, especially if the kids are going to get older. I think the fear there is that you're going to unmask a number of systemic deficits. And working with a number of our families, we continue to believe that that's the case. And so here, we're seeing a durable urinary heparan sulfate, again, 1 year post-administration. You'll note that we will update a bit more as we get to our R&D day next week, and again at the European Society of Gene and Cell Therapy, a little bit more on the cohort 2 data. So then we start talking about some of the biophysical reductions. So I mentioned that many of these children in the lysosomal storage diseases have problems with their livers and their spleens, and they come in and they present with 100 or 200% enlarged livers. So you can actually see and feel the liver margins on these kids that are very big. And so, what the goal of a lot of these therapies is to try and reduce some of not only the biomarkers, but a lot of the biophysical manifestations. And so when we look at this, we look at, again, trying to give this after a single IV administration. You see here that 1 year later, we are seeing a durable effect, a statistically significant durable effect in the liver, showing that and we have made these livers 80% more like normal. Think about that for a second. 80% more like normal. And while we see a lot of the reductions in biomarkers, and we go on and show changes in liver and some spleen changes, what we really want to often talk about is how does this really get to the underlying disease progression for these kids? Now, the mantra in a lot of gene therapy is treat earlier and treat with more. So, I think that many of us a few years from now are going to be talking about products that are trying to treat, 2-month-olds or 3-month-olds, to try and get there and stop the disease progression before it actually begins. So now, as we look to some of our clinical data, we're looking in areas of the brain like the basal ganglia or the thalamus, or the palladium or the putamen, to look at, what type of functions those may have and how a one-time administration gene therapy can modify some of those brain architecture. So again, the AAV9, it does cross. It's the only AAV to date that really crosses after an IV administration. And so, I think that some of the areas where you're looking at trying to drill holes in the skull, have a challenge to try and get into some of the deep structure of the brain, such as the basal ganglia in particular. Basal ganglia is important for voluntary movements. It's important for learning. You see areas of that are important in memory and sensory function. And if you damage those, you have problems. You have muscle tremors. You have motor skill problems. You have impaired learning. So again, the earlier that you can get in and correct the underlying disease deficit, the better that these kids have a chance at some form of normal potential development. So what we see here is that we did MRI imaging over in the natural history study, which will be a little bit more fulsomely reported out soon. But we did see that in the natural history study, a significant decline in the thalamus volume over a 1 year period. And then when we look at some of the kids in the cohort 1, again, these are kids that are average age of about 6.5. We see about 150% improvement over controls, and that's statistically significant. So again, what this is showing is some form of stabilization, or actually a improvement, in the actual maintenance of volume in the brain. Similarly, when we look at that putamen, we're seeing an improvement of about 69-70% over the controls. So again, this is helping show why natural history studies are so important, because they give you things to be able to measure against, particularly with this level of data. So again, looking at an N of 8 subjects in the control group versus 2 or 3 right now in the treated group. So, being able to show that you preserve brain function or brain architecture is important, but really how does that translate to some of the neurocognitive assessments? And so, through our natural history study, we were able to demonstrate that the Leiter exam and the Vineland exam in particular were good assessments, particularly in the kids that were very, very young. So when we first started these trials, we thought we'd enroll between 6 and 7, where these kids are on the other side of that development curve. An important area where we thought if we could see any form of disease attenuation, this might be the area where we might look at, so that when you push potentially younger, you may be able to see an earlier intervention has more benefit. So we see here that ABEO 102 stabilizes actually nonverbal IQ, so the lighter exam at 12 months. I think we were pleasantly surprised to see that this again continued to show a level of benefit in these older children where there has been a significant amount of disease progression. And then similarly, when we look at the Vinland scale, which is the adaptive behavior scale, this is again a significant finding. At 1 year, you're actually seeing an uptick in their behavior, about an 86% improvement over controls from the natural history study. So to date, this is the first and only study in the world that's been able to demonstrate, this level of both biopotency, biophysical, and neurocognitive, whether it's preservation of deep brain architecture or, maintenance or stabilization of some neurocognitive assessments. We didn't really comment on safety. It's been well tolerated over 2,000 cumulative follow-up days with no serious adverse events related to the drug. And we have, of course, additional data on cohort 2 coming out at R&D Day and ESGCT, so very much look forward to presenting that data as well. And so in the remaining few minutes, we'll just talk for a little bit about our EB101 program, which is the recessive dystrophic epidermolysis bullosa. And due to some challenges in this space, this is actually the world-leading program right now for these patients. And so for those of you that are not familiar with this disease, it's pretty much the most horrific disease you've ever not seen, but these children have a problem with the zipper, the Velcro that holds the skin onto their body. And again, they blister, these fall off, their fingers fuse, they're in bandages pretty much most of their life, and on a daily basis have to go through hours of preparation when bandages and bathing to make sure that they don't suffer from infection. It's lethal. Many of the children or many of the adults also die from this disease, usually of squamous cell carcinoma. And so our product is actually, it's a graft. So we take a punch biopsy and we retrovirally correct it, the keratinocytes. So again, one of the important things about this product is that we're actually correcting keratinocytes. So keratinocytes are actually skin cells. So when we put this back on, we're actually giving the patient's own cells back to them that have been corrected. And we see a correction of about 70 or 80% of these cells. And when we look at, this is actually an update on the data. So looking at 7 patients through 6 months, we see a maintenance of, or 3 months of 100% of wound closure. That lasts for about 90% of those patients have greater than 50% at 6 months. So, we brought this to the FDA and they were very supportive and collaborative. Again, they've been very, very safe, and they said, stop your phase 1/2 trial, go ahead and pass go, and actually go right to your pivotal phase 3. So we're very excited to get that news and look forward to starting our pivotal phase 3 in this in the first quarter of 2018. And I'll close with something that we announced yesterday. Many of you in the lysosomal storage disease field or the MPS field know about the Lintons from Canada, but their daughter Alyssa passed last year from MPS 3B. But we opened up in Cleveland and had our groundbreaking ceremony for the GMP facility for gene and cell therapies. This will be our future home for commercial manufacture of our cell and gene therapies. And with that, I'll thank you.